So I'll get started. Hello, everyone. My name is Molly McBride, and as we heard, I am from Texas A&M University, Corpus Christi, and my advisor there is Dr. Michael Wetz. And today I'm going to be talking to you about the influence of river discharge variability on phytoplankton communities in Matagorda Bay, Texas, or as I like to call it, diatom domination, which, spoiler, will come up later when we get to the results section. So to start, I just wanted to make you aware of a progress bar at the bottom of the screen. I have one of my favorite diatoms here in Navicula that'll be hovering over the section that I'm in. So to start, my question was, how does riverine inflow influence phytoplankton? And when I say phytoplankton, I mean how much phytoplankton is present and what kind of phytoplankton is present. And why do we care about this question? So freshwater inflow is incredibly important for estuaries. For instance, it affects salinity, the amount of salt in the water, and freshwater meeting salt water from the ocean is the definition of an estuary. It can also affect temperature, so freshwater inflow increases can help moderate temperatures. And it can also bring in sediment, which is important for the physical structure of our estuaries. It brings in nutrients, which can help fuel production. And finally, it can bring in contaminants, which can negatively affect our estuaries. And this is where phytoplankton come in to get a sense of how all of these potential changes that can occur from freshwater inflow can affect phytoplankton communities. So why are phytoplankton a good choice to look at? Well, essentially, they are great indicators of environmental change. So changes in biomass or species present may tell us something about what's coming into the system. Second, phytoplankton are incredibly important primary producers. They actually account for 50% of worldwide primary production. And lastly, harmful algal blooms, or HABs, are of concern. What does harmful mean? They can be toxin producers. They can physically obstruct organisms, so spiny phytoplankton can get into fish gills and stop oxygen exchange and cause fish kills. And finally, when they're starting to break down, the decomposers that are breaking them down are using a lot of oxygen to do so, and that can cause hypoxic or anoxic conditions. So into my methods, how did we look at addressing this question? We chose a location on the Texas coast that is influenced by freshwater inflow. The freshwater inflow in question is from the Colorado River, which is inlet right above site A1. And we chose 6 sites to capture a freshwater gradient. So A1 is the closest to the river mouth, as I mentioned, and A9 is the farthest away at about 40 kilometers. And the data we collected was we preserved samples to do cell counts, so trying to see how much phytoplankton are there again. And we also tried to identify the taxons that we saw, the taxa that we saw present, and down to the genus level where possible. And we also collected environmental parameters like salinity, temperature, those things that I mentioned can be affected by freshwater inflow. And finally, of course, river discharge, because we wanted to see how riverine inflow affected our phytoplankton. So into the results, I'm going to be talking about some spatial results and some temporal results. So to start with spatial, we saw a clear salinity gradient moving away from closest to the river mouth to the farthest away at site A9. And this is pretty typical of estuaries that are influenced by freshwater inflow. And this can affect the type of organisms that can survive in those areas that have that low salinity tolerance or high salinity tolerance. And we also collected nutrients, and we saw that in all instances, we collected ammonium, N+N, orthophosphate, and silicate. We saw that they all decreased moving away from the river mouth, which indicates that the river might be an important contributor of nutrients to the system. Next, into temporal trends. Over time, we saw that the nitrogen to phosphorus ratio stayed generally below 10, which tells us that the system is consistently nitrogen limited because the necessary amount for phytoplankton to thrive is 16:1. So this is important for the implications of what kind of organisms can survive there with that limitation. And we also saw generally over time that salinity decreased and temperature had a typical regime where it started to increase in the spring and hit its peak in the summer and then started to decrease towards the winter. And now moving into biovolume results. As I said, there was a spoiler that diatoms were going to dominate. And you can see here, I have graphed the diatom biomass in green, and it was actually 65% of total biomass across the entire study at all sampling sites. And here we're starting to see some influence of river discharge. I plotted that below, and you can see that right before that peak in October of 2020, we had a decrease in river discharge. So that may influence our biovolume. And of course, I mentioned HABs, they're of concern. They were actually the cause of closure of this system 6 times. And we saw several potential HAB diatoms, and most of these here are of concern because of physical obstruction, because they have spines. But nichioid in particular is a potential toxin producer, and that's of course of concern. And we in fact saw a bloom of nichioid in November of 2020, and our bloom criteria was above the 95th percentile. And this occurred in November, as I said, and that was right after a decrease in river discharge. So we're seeing the same response in nichioid as the total biomass responded to river discharge. And then in May 2021, we had another bloom of leptocylindrus, and this actually occurred after a huge increase in river discharge. So we thought that was interesting. And in fact, that was the maximum discharge we saw over the entire study period. It was about 3,000 cubic meters per second. So to conclude, we are going to see environmental change, and it's going to affect our estuaries. And one of those changes is, of course, riverine inflow. And this can be caused by anthropogenic changes. So as water use is increasing, as populations grow, we divert from the river to collect that water, and that can lessen the amount that's reaching the estuary. And there's also natural changes in riverine inflow. So for example, Texas is very influenced in rainfall by the El Nino La Nina cycle. And these changes are going to cause changes in primary production, as we saw. We saw some blooms, we saw some increases in biomass. And we could also see a potential increase in HABs based on these changes. So I thought it was interesting that one species, nichioid, responded positively to a decrease in discharge, and leptocylindrus responded positively to an increase in discharge. So we really have to take this by a genus basis to look at the specific species and see how they're reacting individually to these changes. So future directions for this work. This was just a baseline study, and we need to continue sampling so we can track changes over time. And those studies will hopefully be long-term. This was about 1.5 years of data collection, but long-term studies are really going to allow us to capture climatic changes that we can't really see if we only do for several weeks or 1 year of sampling. And we also need to track the frequency of blooms. So are they increasing, and what are they being influenced by if they're increasing in frequency? Also, species identification is very important. We need some kind of technology or an expert to be able to identify down to the species level, because then we can really tell if they are harmful algal blooms that are occurring. And another component about that is toxin analysis. So that nichioid species that I mentioned, it can produce toxins, and it would be good if we had some way of analyzing the amount of toxins in the water to see if that is occurring. So with that, I just want to say thank you, and I'm open to any questions, and here's my contact information. And I wanted to say that this presentation was made possible by the NOAA EPP and also a contract from the Texas office of the comptroller. So thank you.